So first of all, a little bit about BASF as a whole. So agriculture is a component of BASF and is one of the standalone businesses. And if we look in particular, we see that, especially compared to some other of these businesses, R&D is a major component of agricultural innovation. Um, AP, so agriculture itself, is another very broad company. So BASF is a large company. It's sort of a nesting doll of all of these different groups and subgroups, etc. And the specific component of agriculture that I primarily work with is the crop protection unit, specifically with herbicides. Why do we need herbicides? Well, herbicides are one of the management tools that farmers have. Farmers make our food. Food is important, right? Um, Weeds compete with your crops that make food and reduce the amount of food that we get. So weeds aren't necessarily the best thing to have, specifically when you want to make more food. Weeds is, of course, a very broad category. In principle, it's any plant that you just don't want in that particular place could be considered a weed. And if we look from a global screenshot, so this came from the HREC committee of the most prominent weeds, most important weeds across the world, we can see that while there's going to be some weed species that are present across the world, and Ranfus, for one, you will see is relevant in a lot of different countries. A lot of different geographies will have their own special blend of weeds. And to effectively market herbicides, we need to have a management solution available for everyone and relevant to both the cropping systems as well as to the particular mix of weed species that you have available. To make matters a bit worse is herbicides represent a large selection pressure on the agricultural ecosystem. As with any large selection pressure, things tend to evolve to get around that selection pressure. And that is one of the large problems in that once weed becomes resistant to a given herbicide or chemistry, it's very much like antibiotic resistance, that chemistry becomes less useful, but it's also an opportunity because there is then the need to develop new chemistries to meet that need, as well as chemistries that are potentially more resilient against the adaptations that we have seen before or could predict for the future. As you can sort of see, as herbicides began to be adopted, sort of in at least the current version of herbicides adopted in sort of the 60s and 70s, we've had a continuous increase in the number of resistance cases reported across the world, as well as if we look at sort of our major cropping systems, we see that there are a number of species for these major cropping systems which have resistance to them. One of the problems with dealing with such a wide range of species, and as Bjorn alluded to earlier, we don't actually know very much about plants in general. And for the plants that we do know a lot of, they tend to not be weeds. Normally, they're either some sort of a lab plant that's well studied, so Arabidopsis, or some sort of a crop plant, maize, wheat, etc. And that's where the bulk of our knowledge, whether that's in available databases or in the literature, is concentrated on. That's where the money is, that's where the funding is. So we often rely on orthology to try to map this information from our model systems to our non-model systems. This is typically done through some sort of sequence similarity. So genes have sequences. So whether that is your coding sequence or the protein sequence, you can do a similarity matching to essentially say, okay, gene one, and in this case, Allopicuris, is similar to gene one in GMAs. So we're pretending that they are the same thing, and we just sort of transfer the information over and just sort of hope it works. Where things get very interesting is in this scenario, say we don't find a good match for ZMA's gene 3. So that information can't be transferred. So we have a break in the link of information if we're trying to model a certain biological pathway within Ellipticurus. Typically, this is handled either through gap filling. We pretend reaction 3 is there, so the system makes sense and carbon can still flow. But there is also the possibility that there is a different reaction or that both are existing at the same time within Ellipticurus, and thus that adds potential error to our models of these species. If you expand that across sort of the 80 or so relatively important weeds, and if you look closer within a given geography where you have even more weeds, that becomes a bit of a nightmare. Specifically for the information that we're trying to transfer over, most of that information, as with any standard functional annotation of a gene, so what does it do, will typically come from our sort of general standardized databases. Think of your UniProts, for instance. These are high quality, often some level of review, levels of information, but that isn't all of the knowledge that exists. You can think of the flow of knowledge as a sort of how I have demonstrated here, where we have at the far extreme, effectively conference abstracts and presentations or posters, where we have the largest amount of quantity of information, relatively poor vetting of information, and it tends to be very, very specific, also potentially very specifically relevant to our non-model systems of interest. Some of these, not all, will eventually make their way into some sort of a paper. Even few will be openly accessible. From these papers, they can be accumulated into community relevant resources. So we have here Tear, Soybase, and the maize database. And from these community databases, sometimes automatically, sometimes more manually, this information also gets propagated to UniPro. As all of this information propagates to our highest quality data sources, we get less and less of it passing the filters or just not getting caught at all. And this information tends to become more and more generalized. So you can imagine if you look at a UniProt description, you will see maybe in a short one paragraph to a few sentences that was derived from maybe 20 papers. So perhaps there is also a lot of nuance that is being missed that may become important when you want to develop or test a hypothesis. So we have a clear need, which is to try to consolidate the biological context from these knowledge-rich systems, relatively speaking, knowledge-rich systems. It could very well be very knowledge poor if you compare it to something like human or a more defined, smaller system, and use this to transfer to knowledge poor systems and try to make sense and address the needs of our particular partners. The current status quo is your standard annotation tools. So you do a homology mapping to, say, UniProt, and you take the closest match or maybe the few closest matches and provide those short descriptions as well. This is what the gene probably does. Integrating the various data across sort of the knowledge spectrum is very important, as well as integrating that public data within our own internal discoveries. So if we invest in finding out what this gene does, either in the context of a herbicide or in the general context of a plant, we want to be able to use that in the future through sort of integration into these databases and making it available broadly. This has the added benefit of improving the AI readiness of the data. So AI is one of the big buzzwords across the globe, right? And you've heard from the past previous talks a lot of utilization of unstructured data and literature to address these. And in this case, this is also something that we need for the to represent and access and query these sort of knowledge-based systems, as well as making the data relatively fair. The slightly marketing-driven term we used to describe this was a plant twin graph, where we're trying to aggregate and connect the various sources of information in a biologically relevant way, ranging from sort of just protein straight off of a genome annotation, maybe some papers, some of our endpoint data phenotypes, and some of our measurements, various omics data sets into a single data integration, in this case, a Neo4j graph database. Hence why I was invited to give this talk. The process started very recently, at least for us. So it started September last year with sort of the kickoff of this project. This was in collaboration with the Neo4j Professional Services. My background is as a geneticist, so less of a machine learning guru, less of a large data management integration guru, but very much coming from more the end user side of it, having picked up a lot of bioinformatics experience over the years and trying to build these systems from more or less scratch. One of the two of the major challenges, as you've heard before, one of them is identifier mapping. So a lot, especially when you're diving into literature, you get into a situation where, depending on when the paper was published, they're going to be using a different version of the genome that may not necessarily be compatible with the present version, as well as you can get into specific differences amongst accessions and how the different identifiers can be reported. This is a problem across all of, at least, plant research, as well as considering that internally also we're going to have our own internally developed genomes for the organisms we're interested in, and how do we match those identifiers in an intelligible way. And the primary project ended February this year with sort of the use case evaluation, defense to the innovation groups, and sort of the continual process in all big companies of trying to convince people to take the next step. As mentioned before, data integration is not a trivial task, right? You've heard not just me, fortunately, complain a bit at length of identifier mapping, just in the genomic space alone is a bit of a problem, but also when we get into integrating different databases together, they can have different core identifiers that may not necessarily be compatible. So for instance, how do you make a broad EC number compatible with, say, a group of genes related by sequence ontology? This brings us to our developed core data model. So here is a very simplistic representation of a biological system. We have our chemical reaction metabolic network. We have your classical gene encodes transcript encodes protein. We have some information storing nodes such as annotation and orthology nodes to mediate connections both to free text fields, more or less, as well as to and amongst other species. To this, we also integrate experimental data based off of the developed endpoint or developed measurements that we can get. So chemicals you can measure through some sort of a mass spec approach. Genes can have different variants in them. Transcripts can have differential expression in them. Then you have your study and experimental metadata stored in there, because that is what allows you to do cross comparisons amongst experiments and amongst studies. Going to a practical example, I had to think a little bit to find something I could actually talk about. Here we're talking about dwarfing in lemna. So lemna is a small aquatic plant that you see on the surface of ponds, per se. There's a paper from the mid-2010s, I think, talking about what we use Lemna for. And here we have a series of 5 data sets that are used to populate a small graph. We have sort of a central reaction network. This is a subset of it where you have relatively short path leading to a large differentiation of potential molecules. We have some literature-derived phenotypes. These ones are from Arabidopsis. This is pretty much a free text field that says plant got shorter, for lack of a better term. We have our orthology groups to link our Arabidopsis to our Lemna, and we have the genes from the various genomes themselves. So to give a little context as to what this could be used for, say you want to either make Lemna shorter or have some sort of an external stimulus to affect either the production of these downstream products or to have like xenobiotic treatment to make the plant shorter, right? You would look at this reaction network. There's a number of potential choke points. Literature has declared, or other people have found, that interrupting these choke points with loss of function mutations or knockouts results in the phenotype you're interested in. One of these, in fact, the one with the most literature references to it, has had a gene family expansion present within the Lemna. So instead of just trying to interrupt one gene, you're going to have to try to probably interrupt 5 genes. Makes it a bit less of a good target. So maybe you'd go for one of these other genes as your target with 2 or 1 literature citations. You can then imagine expanding that not just for one plant, but across potentially 80 or so different weed species to aid in your target identification tasks. Going to another use case is in, of course, the annotation task. So what does random gene ID do, for instance? We can hook our graphs up to our various agentic approaches and ask a relatively simple question. What does this do? Give me a functional annotation of this gene within the context of this graph, right? And here we have a comparison of sort of a graph-mediated summary just based off the annotations associated with proteins within the orthology group, as compared to the information we get from our standard annotation tools, whether that's InterProScan or Eggnog, another such tool. Eggnog using a very similar conceptual approach, though not using a graph. And you can see we can get at least a more verbose description, as well as, importantly, the ability to ask further questions from within the given context. So for instance, I want to also include some BPI and reaction network information. And here I get, okay, we have some metabolites that are being converted into, as well as some more global context of what is, in this case, downstream. Don't know how I am doing for time, but this is my final slide. While it's a 3-person month project, there were a lot of people involved in it, both from the Neo4j consultancy team as well as from the core group. So myself, Avreen, and Vidya did a lot of the data wrangling and thought together with Martin and Stuart. Agentic systems from Elliot and Kuhn, as well as a number of people involved in sort of the software and infrastructure support. Getting this working, getting a new thing working within existing systems is always something that requires a few hands on hand. With that, I do believe I am done.